Love the enthusiasm. Okay, so what is lymphatics? Like, what's this exactly? Well, I'm sure you're all familiar with the cardiovascular system now. So, blood is distributed from the heart outside to the systemic and pulmonary circulation by the arterial system, and blood is returned by the venous system. All right, so the heart, located in the thorax, the arterial, oh, this is interesting. The arterial system, we can divide it into the aorta. It's gonna go down into large arteries, branching down into smaller arteries, until we reach this tissue level that we're calling the capillary beds. So at the capillary beds, that's between arterioles, intervenules. So what are capillaries? Capillaries are essentially these cylindrical tubes that are made of overlapping endothelials, a single layer of simple squamous cells. So they're super thin, fluid's gonna rush past them. And what's happening when you've got this somewhat porous tube with fluid coursing through it? That's right, you're gonna get some fluid leakage. And what's in here? The liquid components of blood, water and solutes, are going to leave through the tiny pores between these cells. So you're going to get kind of like this filtration effect almost. Now, if blood is leaving, the fluid compartment of blood is leaving through capillaries, where's it going to? That's right, it occupies the space right outside the capillaries. So we've got a super special name for the space. We're calling it interstitial space. So that's the space between your capillaries and the outside of the cells. So fluids essentially, as you know, the hydrostatic pressure, the fluid pressure of blood is coursing through your capillaries. It's going to squeeze out this fluid out into the interstitial space. Alrighty. So let's do some quick math here. I'm not a math person, and you're not expected to remember this, but just for illustration, every day you're sending about 20 liters of blood from your capillaries to your interstitial space. Now, about 17 liters, it's going to get directly reabsorbed by your venous system. Your venous system does a great job at like sucking it right back in, but that leaves about 3 liters of fluid in your interstitial space. Now, I'm a metric person, so 3 liters makes sense to me, but that's like 2 cans of soda pop. So 2 cans of soda pop or the liquid just sitting about in your extremities, yeesh, that doesn't sound too good. And that's where our lymphatic system comes in. Here's a little roadmap of what we're going to be doing today. We're going to be discussing the function of the lymphatic system. We're going to be looking at a little bit of immunity, and we're going to be discussing the organs associated with the lymphatic system. Alrighty, so the cardiovascular system is kind of circular, but the lymphatic system really isn't. It's just got one direction. One direction, that's funny. So what it's responsible for is moving fluid from your capillary, your extremities, directly into the venous system. That's all it does. Right. So let's go back to the 3 liters that we were talking about. The three is going to be returned by your lymph node. Woohoo! And that we're going to be discussing as the primary function of your lymphatic system. It's the regulation of fluid, the fluids in your bloodstream. Now, we're not going to want all of this fluid out in our interstitial spaces. That's going to result in a type of swelling that we're calling edema. Again, we're also not going to want all of this fluid leaving our body. That's going to leave a critical mass of fluid missing from our blood. So we call that hypovolemia. Alrighty, so what are these lymphatic capillaries? Let's take zoomed in look We're looking at overlapping endothelial cells again, but now these are kind of wonky in how they're arranged. They've got kind of they're set up in a way that they act almost like one-way valves. So we've got interstitial fluid right outside. It can get inside the lymphatic capillaries, but once it's in, there's no way but up. It's gonna go right up completely once it's in. And now, when this interstitial fluid is inside the capillaries, it's gonna get a new name. Rebrand. We're calling this lymph or lymphatic fluid. It's essentially just tissue fluid, but now it's inside your lymphatic capillaries. So we're giving it a different name. Woo-hoo. Okay, so let's look at the actual capillary. This is the wonky overlapping endothelial cell structure that I was talking about. As you can see, they're kind of like sitting on top of each other. And this configuration gives us some interesting structures internally. Now I'm taking a longitudinal slice across this lymph vessel, and you're seeing, well, you will see things that are almost like one-way valves. Huh. Now, where might have we seen this before? Do we remember? Do we remember?
Veins.
Veins. Yep. Great job. So, why do veins have these one-way valves? Again, you're gonna want to prevent blood from pooling down. Same thing here. We're gonna want lymphatic fluid moving up against gravity. Now, if you might recall, for the circulatory system, you've got a central pump, the heart. For the lymphatic system, however, you don't have a central organ that's pumping everything up. You're on muscle movements and pressure gradients that are created by respiration, for example. So this one-way valve becomes super important. Yep. And this is kind of an illustration about how fluid moves up, can't really move back down. Okay, there we go. Yep, goes up, no way back out. Now, what is this ubiquitous lymphatic fluid? It's mostly just water, 92% water, and 8% of it is composed of a bunch of other stuff. There's ions, electrolytes, all your little salts, proteins, and most importantly, some metabolites. Now, what are metabolites? These are kind of products of cellular activities. You've got carbon dioxide, you've got ammonia, and a bunch of lymphocytes. Now, this composition might sound super familiar. That's because it's basically just blood plasma with a little more waste in it. And that brings us to our secondary function. We're transporting nutrients, hormones, and waste via the lymphatic system. So in that regard, it's almost like the back rooms of the circulatory system really is. Again, okay, what do I mean by back rooms? Yeah, it's kind of shady. It's larger than your systemic arteries and capillaries, so you're gonna be able to move a lot of stuff that you typically can. So it is literally the back rooms. Okay, so we're gonna take a hard pivot, digestive system. You're gonna cover this with Dr. Rowe later. We're zooming into a section of the small intestine. If you zoom really closer, you get to see these tiny little bumps. They're called villi. And if you zoom again into these villi, they've got even more tiny projections called microvilli. Now, what do you see? You see a vein, an artery, and gasp, a lymphatic capillary. Now, these are very special lymphatic capillaries. They're called lacteals, and they're going to help absorb lipids from your diet and turn it into this milky white fluid called chyle, chyle, not the name, chyle, and transport them around the body. So that's another example of our secondary function, absorption and transportation of lipids in this context. Because lipids are large and they're binding into chyle, CH, and transport them. Yay. Now, very closely associated with our digestive system is another organ that sits right behind your stomach. It's this reddish brown organ that we're gonna encounter soon. We're calling it the spleen. Now, if you were paying attention to Nora's lecture, which I hope you were, what does the spleen do? It's gonna remove damaged RBCs, and recycle them. We're gonna take the heme and the globin and recycle them to make newer RBCs. The lymphatic system in general is gonna, in some way, be responsible for removing damaged cells from circulation and reusing them, recycling them. It's also responsible, since we've got all of these cells that do phagocytosis and are responsible for other immune responses, your lymphatic system really important in blood and other fluids for antigens and pathogens and making sure that everything that's in there is supposed to be there. So yeah, let's recapitulate what I've discussed so far. What are the functions of the lymphatic system? We've got maintaining the blood fluid volume because we don't want edema and we don't want to be hypovolemic. We've got transportation of nutrients like bile, or hormones and cellular waste back into your cardiovascular system and across the body. We're also removing damaged cells from circulation and recycling these cell fragments because we've got all of these lovely cells that do phagocytosis. And we're surveying blood and lymphatic fluids for foreign cells and antigens. Lovely. So we're gonna do check. Yay. Okay, now let's look at the organization of this system. Your lymphatic system is essentially your circulatory system, multiple levels of organization. The smallest level that I was talking about are these little things over here. These are your lymphatic capillaries. So your lymphatic capillaries are going to be the smallest level of organization. Again, you're not going to see any valves over here, but these are still one-way portals. Now, a bunch of your lymphatic capillaries are going to come together, organize, and form a larger structure. Now, this is your lymphatic vessel or your lymph vessel. Now, it's just a bunch of capillaries that come together to form something bigger. And now that it's bigger, you're gonna start seeing these structures, your internal valves. You see these at lymph vessels. Now, if we're building up, a bunch of lymph vessels are going to join to form a lymphatic trunk. And again, these are responsible for draining a particular body region, for example. Now, one good example for a lymphatic trunk is all of this in your small intestine. Now, all of these lymph nodes are going to drain chyle, and this is the only lymphatic trunk that you'd be expected to know. For this class, we're calling this cisterna chyli. It's a fun little collection of lymphatic vessels merging into a lymphatic trunk that are all responsible for collecting chyle from dietary fats. How lovely. Okay, now, all of these trunks that are draining different body regions are expected to drain into one of two collecting ducts. Now, there's two of these, one on each side, and they're super predetermined for what body regions they drain. Now, you've got essentially the right head, neck, chest, and right upper extremity being drained by your right lymphatic duct. Now, everything else, so we're talking left head, left neck, left upper extremity, your abdomen, your lower extremities, your pelvis, all of that's going to be drained by your thoracic duct. Your thoracic duct is also one of the structures that you can actually observe in lab. So when you're in lab, you're going to want to look for the left subclavian vein and kind of just like look behind it for a thin filamentous structure. That would be your thoracic duct. Super thin, super fragile. Not sure if it's going to be super well preserved, but if you see it, bam. Okay, so why do we care about lymphatics? Now, if you're familiar with any form of looks maxing, you know that there's this technique called gua sha. And people on TikTok, they're going to claim that it's magic crystals that absorb the puffiness from your body. Um, Magic crystals, kind of doubt it, but it is lymphatics. So, you've got facial puffiness that's a mild form of edema. That's just tissue fluid collecting in your face. Super normal. Now, if you want to resolve this puffiness, you're gonna want to move this lymphatic fluid out of your face. Now, how do we move it out of our face? Do we move it up? No, we're gonna move it down towards our neck. And that's why we've got this cold stone that's essentially being used to shift lymphatic fluid down and out into our cervical lymph nodes so they can be drained into your subclavian veins. And that's how a gua sha works. Okay, so now we've talked about the function and the organization of the lymphatic system. Woohoo! Look at you go. You're learning. Okay, now let's talk a little bit about the immune system and the anatomy that's pertinent here. Again, this is not an immunology class, so if things sound a little too simplistic, that's all you need. So let's establish some ground rules over here. You've got cells that belong and that don't belong to your body. Simple as that. Now, these immune cells are from us, the host. You've also got pathogens. Now, these cells are foreign. We don't like these. Our body likes keeping its own cells safe. And how does it know if a cell is foreign? Well, it's gonna look for these tiny little surface proteins that are these red dots over here that are antigens. Now, your body has a detailed directory of all of its own antigens. So when it encounters an antigen that it doesn't recognize, it's gonna launch an immune response. Now, how exactly does it do that? Now, your body is gonna make these complementary structures called antibodies. Antibody. And the cells that make them are B cells. Get it? Because body, antibody. I know. Super fun. So these antibodies are secreted into circulation and they're freely flowing, just looking for its antigen so it can destroy it. How lovely. And that's essentially all of the principles of immunology you need to know for anatomy. Lovely, isn't it? Yeah. But how do cells know who we're attacking? So, how do cells talk to each other, essentially? Well, we've got this interesting idea that we're calling antigen presentation. Now, it's exactly as it sounds. We're taking foreign antigen and we're presenting it. Immune cells of the body. Yeah. So, let's take this pathogen, for example. You've got a class of cells. Now it's a subcategory of cells that we're calling antigen presenting cells, APCs. You've got two major categories, two major cell types that can function as APC cells that present antigens. You've got dendritic cells, and you've got macrophages. So what these cells are going to do is that they're first going to ingest this enemy cell. They're going to eat it up. They're going to break it down. And they're going to take fragments. They're going to take surface proteins from this foreign cell and present them on a surface. It's kind of like parading around with the heads of its enemies on pikes. So it's essentially putting out a hit list for these cells. Sounds gnarly, but it's pretty much what's going on. So yeah, once you've got all of these fragments of antigens being presented on the surface, your cells are just gonna circulate across lymphatic tissues. So they're just gonna, yeah. Now, the cells that are traveling, your traveling cells, are T cells. T for T, traveling cells. I know. That's not why they're called T cells, but hey, whatever helps us remember. Okay, now let's look at our first subcategory of antigen presenting cells. We've got macrophages. Now, these might sound familiar. Do we remember learning about monocytes? Now, what are they? Shout them out. What do we remember about monocytes? We could be here all day. Yes, it is a subcategory of leukocyte, a white blood cell. What do we remember about its granules? Well, they've all got granules, you just can't see these. So, despite it being an agranulocyte, still has those granules. And now these granules in specific, they're gonna help the cell perform a interesting function called phagocytosis, eating them up. So it's kind of like a janitor that just picks up trash and eats it. Yeah. It's like, oops. Your macrophages are super adaptable to whatever tissue. Because remember, they move out of the bloodstream and into body tissues. You've actually seen, would you believe me if I say you actually know a bunch of macrophages already? Well, you do. Do we remember osteoclasts, the cells that break down bone? They're modified macrophages. Those are macrophages so suited to the bone. We've also seen these things. I'll give you a little hint. We're looking at nervous tissue over here. So you've got neurons in red, and these tiny little green things, those are microglia. Those are just macrophages that are adapted to your nervous system. You'll also see a bunch of these in your respiratory system, which is coming up. Wink, wink. So yeah, as we remember, these are agranulocytes. They've got super translucent granules. You just can't see them. And monocytes and macrophages. Let's look at the other category, your lymphocytes. Now, your lymphocytes are the most abundant cell in your lymphatic system. They're also the most abundant white blood cell. Now, one way to identify them histologically is that their nucleus is going to take up about all of their cell volume. They've got super, super chunky nuclei. Now, you've got three different types of lymphocytes. You've got T lymphocytes or T cells, B lymphocytes or B cells, and natural killer cells. Now, I'm going to discuss natural killer cells first because they're a lot simpler in terms of what they do. Now, these cells are your blind killers. They do not care what they're targeting and how they're targeting. They're just gonna go in, fight, and eat them all up. Now, what do I mean by this? These are not antigen specific. These are not trained to target a particular cell. These are essentially weapons of mass destruction. Now, your other two cell types, you've got T lymphocytes and B lymphocytes. Now, T, these cells are gonna travel to your site of infection. Your T lymphocytes are essentially trained to target one particular type of antigen, and they're gonna go out in body tissues and body fluids, seek for these antigens, target them, and yeah, direct cell to cell killing. Now, this direct cell to cell killing has a specific name. We're calling this a cytotoxic response because it's toxic to cells, cell to cell killing. Now, your B lymphocytes, on the other hand, they work from home. Uh, these B lymphocytes are gonna reside in your lymphatic tissues and they're gonna manufacture antibodies. Now, your antibodies are kind of like these tiny little fragments that go out into circulation and tag a particular antigen. Now, these are also antigen specific. They're going to essentially seek out the antibodies, a particular protein, and clump onto them and essentially mark them for destruction by other cells. The key point that I want you to remember is that B cells are not mobile. They stay in one place. They work from home. And I've got a cool immunofluorescent image over here. Here you can see your T cells performing a cytotoxic response. So you've got you've got T cell over here, and it's eating up the cell that wasn't green earlier. Now, your B lymphocytes, on the other hand, we're looking at a lymph node. Now, these things over here, these are its antibodies. It's just secreting them out, and your antibodies are going to go out in the bloodstream and do all the work. Now, if you're like me, like a year and a half ago, and you're like, okay, I'm too scared to ask what the T and B in lymphocyte, what does it mean? Why are there two different cell types? Don't worry, I got you. That's what we're going to be discussing next. Now, depending on what part of your body they're achieving maturation in, your lymphocytes are of two types. Now, let's go all the way back and talk about your hemopoietic stem cells. Now, these are stem cells that can pretty much become any type of cell in the bloodstream. Now, your your hemopoietic stem cell, it can become a red blood cell, can become a white blood cell. But in order for it to become a site, it must commit into becoming a lymphoid stem cell. Now, once it becomes a lymphoid stem cell, it can only become one type of lymphocyte, either a B lymphocyte or a T lymphocyte or a natural killer cell. Now, this whole commitment process, it's kind of like an initiation ritual almost. This happens in the red bone marrow. So we're thinking flat bones, kind of clustered around the axial region. So once you've got these lymphoid stem cells within the red bone marrow, they can essentially just mature on site into natural killer cells or B lymphocytes. Now, B as in bone marrow, that's neat. Now, the other possibility is for your lymphoid cells to, they're gonna migrate to the thymus. Don't worry, we'll discuss these pretty soon. And within your thymus, they can mature into T lymphocyte. They're traveling cells, T lymphocytes. And they attain maturation in the thymus. Wow, that makes sense. Okay, So this whole process of how they attain maturation, we're calling this process immunocompetence. And I'm just gonna plug my computer in because Zoom's draining my battery. Give me 1 second. How's everyone doing today? Are we doing amazing? Am I confusing you? No one wants to talk? Damn. We're just gonna, yeah. Yay. Okay, so let's look at this red bone marrow. Where would we find red bone marrow? So now, when you were a child, many, many years ago, uh, all of your bones had some form of red bone marrow. But as an individual ages, a lot of this red bone marrow, especially in your extremities, is gonna convert into another form of bone marrow called yellow bone marrow. Now, this is more latent, and it's basically just a storage site for adipose tissue. When you're a skeletally mature individual, that's to say about 25, so your mid 20s, all of your red bone marrow is going to be clustered in your axial skeleton. So we're thinking within the bone marrow in your cranium, your ribs, your vertebrae, pelvis, and the proximal ends of your proximal long bones. So the head of your femur, the head of your humerus, etc. Now, now that we've discussed bone marrow, let's the organ that I mentioned. Okay, so we're peeking into the thoracic cavity and we're looking at this one particular space called the mediastinum. And there's a humongous organ over there that's kind of important. It's called your heart. So superiorly to your heart, still in the mediastinum, you're gonna see a blobby structure. This structure that almost looks like a blob of is your thymus. And your thymus, it's a bilobate organ. So there's two big lobes, and it's located in your superior mediastinum, so right about your pericardial cavity, your pericardial sac. And it's super, super active in childhood. And like most, yep, right over here, as you can see, your thymus is the largest when an individual is a newborn. And then it's just gonna slow down, disintegrate, and eventually, so when you're looking at donors in lab, you're probably just gonna see like a bunch of fat that once used to be the thymus. That's pretty common. Okay, now let's look at histology. Who doesn't love histology? I know. But there's a few easy things that you should be looking for. There's a few key features to identify your thymus. Now, your thymus is going to be divided into lobules. So those are these individual island sort of things over here. And these lobules have a distinct color pattern. They've got two main regions. They've got a outer cortex that's typically darker and an inner medulla. Now, as you remember, the thymus is the place of maturation for T cells, T lymphocytes. So it kind of makes sense. So from your medulla, your cells proliferate outwards, or they turn a little darker. Alrighty, those are your primary organs, the organs that are actually producing these lymphocytes. You've also got secondary lymphatic organs, your peripheral organs. Now, one major large organ over here, it's the spleen. your spleen is a reddish brown structure. You're gonna find this posteriorly to your stomach. And in a lot of dissections, you might be tempted to be like, oh, it's behind the stomach. Oh my God, it's the kidney. Not. It's just the spleen. So yeah, it's got this dense connective tissue capsule that's this reddish brown color. And there we go. Capsule in specific, first capsule that's gonna invaginate. It's gonna essentially dig into the tissue layers and form septae. Now, these septae, these are super important. These septae, they've got a specific name. We're calling these trabeculae. Now, your trabeculae are gonna bring in blood vessels in and out of the spleen. Again, because you've got these little partitions, it's gonna divide the spleen into lobules again. Okay, we're looking at a histological section of the spleen, or we will be looking at a histological section. There we go. Now, what's one striking thing that you notice? What's the difference between your thymus and your spleen? What do we have and what do we not have? Do we see individual islands? No, it's all just a huge aggregate with a bunch of sinuses over here. Now, this aggregate, you're seeing specific patterns over here. Now, each of these lobules has two different types of pulp. You've got red pulp and white pulp. Now, the white pulp is not exactly white because you're staining it and the stain's purple. So if something's white, it's going to stain purple. I know, mind-blowing concept. So yeah, all of this over here, the purple, that's your white pulp. It's gonna contain, you guessed it, white blood cells. Your red pulp, on the other hand, is responsible for recycling red blood cells. Okay, now, I've got a little clinical correlation for y'all, and I'm gonna want you to pay attention because you, yes, you, are a medical student in the future on a trauma rotation. There's a 24-year-old man who's brought to the emergency department after a high-speed motor vehicle collision, and he was a restrained driver. This individual is awake, he's alert, he's answering questions appropriately, but he is annoyed more than anything else. And he keeps insisting that he's basically fine and that he wants to leave. On the airways patent sentences. He is a little tachycardic, but that's fine. Blood pressure is like 120 over 80. Respiratory rate is 20. Everything's fine. But there's this little brooding going on their abdomen. Because he's still hemodynamically responsive and, sitting up and talking, they're rushing him into a CT scan. Yeah, while being transported and prepared for imaging, he becomes a little paler, a little restless, and he insists that he wants to go home and suddenly goes quieter. Heart rate's going up and blood pressure's falling. And moments later, he's on the floor, collapse. Imaging is revealing a splenic laceration with hemoperitoneum. Now, what's going on? What just happened? He was fine. He was up talking like 20 seconds ago. Oh, lovely. We've got his CT. Now, I've got a normal CT off here to the side to show you what you typically expect to see in an abdominal CT. Now, your spleen is right over here. Do we see these well-defined borders? Do we see well-defined borders? Yeah, that's what I'm expecting to see. But right over here, do we see well-defined borders? No. That's essentially because remember the thick fibrous capsule that's keeping all the pulp in? Well, that capsule is being lacerated. And if a degree of laceration is a little too much, we can get a condition called a splenic rupture. Now, what do we think was the cause of this splenic rupture? Remember, this individual was a restraint driver, so we're thinking seat belts. Now, um, I'm not your mom, but there is a correct position to place your seatbelt and so many incorrect positions. So your seatbelt, you're gonna want to place it over your sternum, not by your neck, not by your stomach, and your arms are gonna be under, not over. So yeah, wear your seatbelts properly. Okay, that was a nice little tangent. We're gonna go back in and look at lymphatic tissue in a little more detail. Now, we've got other kinds of secondary tissues, lymphatic tissues, that is, and we're calling these MALT. Ooh, what does that stand for? Mucosa-associated lymphatic tissue. Now, this is nothing super foreign. This is just lymphatic tissue and mucosa. So you've got connective tissue and reticular fibers. There's a lot of capillaries because you're going to want innervation. And you've got two major regions, nodules, a primary and a secondary. Now, these nodules are what's going to help you identify whether you're looking at a lymph node in lab, for example. Now, depending on what part of the body these lymphatic tissues are in, they've got different names. You've got GALT, gut-associated lymph tissue. You've got BALT. You've got Peyer's patches, not SALT. I know, missed opportunity. Now, one specific type of MALT are your tonsils. Now, your tonsils are this mucosa-associated lymphatic tissue that's specifically along the pharynx. So we're thinking your airways and yeah, your airways and your the lamina propria. So we're thinking the layer underneath the mucosa, it's going to contain these mucous membranes with the lymphatic tissues in them. Now, these are going to do a little invagination. So you're going to see kind of like these pits. These pits are called crypts. These are going to trap in pathogens and bacteria and activate these nodules. And these tonsils are set up in a way that it forms a circle of immunity around your pharynx. You're going to look at lymph nodes. You're going to look at tonsils again in GI. So I'm not going to go to that in too much detail. But yeah, lymph nodes in general, though, you're going to find them in clusters. So when you're in lab, you're going to find lymph nodes aggregated along certain sites. Now, lymph nodes are these structures that are going to filter lymphatic fluid. Now, these are the ones that are doing this by filtering lymph. Okay, we're thinking afferent vessels that arrive at the lymph node, they're bringing lymph into the lymph node, and efferent vessels that are taking it out. Now, each lymph node is going to receive a lot of afferent vessels, and they're all going to be by your convex border. So we're thinking the part that bounces out. Now, your efferent vessels, the exit's always going to be concave border, the part that so as filters lymphatic fluid, it's gonna encounter these regions. It's gonna encounter an outer cortex, a paracortex, and a medulla. So your outer cortex, now this is where your B lymphocytes are living. This is where they live. Your paracortex is where your bloodstream brings in these traveling cells, your T lymphocytes, and your medulla is basically a system of anchoring cords because it's got reticular fibers. Remember the fishnets that trap cells? Okay, we're just gonna look at our outer cortex for a second. We've got primary nodules and secondary nodules. Now, don't get too hung up on this. How do you differentiate these, especially in lab? Well, your primary nodules, you're going to see a big dark blob in tissue. Now, what is this dark blob? Primary nodules are called primary because there's no germinal center. These are just undifferentiated. These are essentially like a lump of clay just sitting up there. Now, your primary nodule, under right conditions, can become a secondary nodule. Now, how do you identify a secondary nodule? You're going to look for a germinal center. Now, a germinal center is kind of like a light patch in between the dark patch. So it's kind of like concentric circles. That's how you know that B cells are maturing and moving outwards. So yeah, patterns to identify. Well, that's all I have for you today, Divas. I know you're gonna do amazing.